we asked our first poll question right at the top of the hour, and we have our results back, and perhaps that will help us shape this conversation a little bit more. Looks like about just under 50% of our audience today are at the translational and early stage clinical trials, so phase 1 and or 2A, followed pretty closely by preclinical. We do have some mid to late stage clinical trials with us today, so that's really good information to know as we kind of carry on the conversation here. And so, Donna, I'd like to talk to you first a little bit about the autologous batch-to-batch variability. So let's talk a little bit about what are, in fact, the biggest challenges in batch-to-batch variation for autologous cell therapy manufacturing, and why?
From my perspective, it goes back to two aspects. It's understanding the incoming seed material. I don't require, but we ask of all the apheresis collection centers to give us a pre-CBC on the subjects, as well as the CBC on that leuk. And we get that in advance of actually receiving our leukapheresis product. And it allows us to understand that hematological composition. It's a little bit more difficult to get the pre-treatment information on the patients, but we know what our incoming seed material is going to look like. And over the course of time, we have developed specific processes, modifications to manufacturing. If we see this, then we do that to give us our desired actual culture material that we really want for the actual manufacturing process. That helps tremendously. Being able to implement it, and an example is if we see an increased number of immature red blood cells, our particular culture system happens to like to grow those. So we have a process in our processing of our leukapheresis that will remove those immature reds, and then we get a more desirable product to go into the actual culture process. The other thing that I think is hugely important is very, very early introduction of automation into manufacturing. You can train, and the more manual process you have, the more deviations you're going to get along the course of time. It's a little bit more different to be consistent. And it's this day and age for cell and gene therapy, there's a lot of automated processes that can be incorporated at the various stages of manufacturing. So early integration of that, I think, is important. It helps to reduce that variability and give you a more consistent outcome. The other thing that does also is it minimizes those very expensive and time-consuming comparability studies that you may have to do if you delay a long time in introducing automation into your manufacturing process.
Yeah, fair enough. And that's really good information too. We, same thing, like I said earlier, we cover automation quite a bit. And we hear exactly what you've shared from our subject matter experts about the need for automation and the need to make the manufacturing processes even more automated as we go. Omkar, I want to give you the opportunity to answer that question as well. To reiterate, what are the biggest challenges in batch-to-batch variation for autologous cell therapy manufacturing, and why?
Yeah, absolutely. And I know you specifically asked about autologous cell therapy, but the same challenge applies when your starting material is not necessarily the apheresis product from the patient, but it's also a tumor biopsy, right? We have the first TIL therapy that was approved not too long ago, less than a week ago. We have other types of personalized cancer vaccines that rely on a patient's tumor biopsy. So there is variability in the way you collect the biopsy, in the way you process it. And so the one thing we must all realize, and I'm sure our audience is also in agreement, is that there is going to be some inherent variability in this. You're dealing with a biological product, you're dealing with a patient-specific specimen. So yes, there is going to be inherent variability that you cannot control, right? But there are so many more aspects, the way, as Donna mentioned as well, there's so many more aspects of how we can control or standardize or harmonize the operational aspects of how we deal with these tissues, how we push these through manufacturing processes, because the biggest impact of any of this variability is that you get a batch that is out of spec or you have a failed batch, right? And the so what is that? One is you have a dose that cannot be used for the patient. Two, in a commercial setting, it's lost revenue for the manufacturer, for everyone. So you want to try to, the biggest loss of all of this is that the patient is not being dosed. The patient who's waiting on the other end of this journey is not receiving his or her dose after having waited for several weeks. So that, going back to your question, those are some of the biggest challenges when it comes to cell therapy manufacturing, where you're sourcing a tissue from a specific patient.